This is video number 3 of protein structure and function. In this video, we'll talk about proteins that function as multi-subunit structures. We'll also talk about interactions between a protein and its binding partners, its ligands. This will include a discussion of enzyme-substrate complexes and how they work. We've talked about several levels of the structural hierarchy of protein structure already. For example, you already know that the sequence of amino acids in a polypeptide chain would make up its primary structure. The secondary structure consists of local folding characteristics as neighboring amino acids interact with one another. At this level, we looked at alpha helices and beta sheets. The tertiary level of structure would involve the folding of an entire polypeptide chain. So it would be those global folding characteristics that hold it together. Now, many proteins consist of one single polypeptide chain, and so the tertiary level of structure would be its highest. However, other proteins are multi-subunit, and so they consist of several polypeptide chains, each folded in its own characteristic manner, and each one of them interacting with the other subunit or subunits in specific ways. That would be its quaternary structure. Those proteins that do their functions as a cluster of folded polypeptides have some interesting characteristics. You may already know some examples, like, for example, the octamer of histones that makes up a packaging unit for DNA in DNA chromosomes in a nucleus. Hemoglobin, which transports oxygen in the bloodstream, consists of a tetramer or 4 monomers. Now, the monomers of a multi-subunit may be identical to one another, or they may be different from one another. But the way they bind with one another is unique, and each subunit may bind to the other in the same way or in different ways. If we look at these two examples here of binding sites, on the left, the cap protein is a dimer formed by interaction between a single identical binding site on each one of the monomers. So the two monomers are identical to one another and their binding sites are identical. On the right, however, this tetramer that you see consists of 4 subunits that themselves are identical, but the binding sites between them are non-identical. And so in the schematic at the bottom of the page, you see that they each have one binding site illustrated in blue and another one illustrated in orange, and the blue and the orange binding sites interact with one another. The example on the screen is the oxygen transport protein hemoglobin. Hemoglobin consists of 2 dimers that are joined together to make a tetramer. Each of the dimers has an alpha chain and a beta chain, and the fusion between the alpha and the beta has a unique binding site for each of the dimers, and the fusion between the beta chains and between the alpha chains in order to make the tetramer are also unique. Sometimes protein monomers assemble into very large functional complexes consisting of many, many monomers. These functional complexes may assume the shape of a filament, they may form sheets or tubes, or they may form spherical structures. This figure 4-27, illustrates what we talked about. The structural protein actin is formed by a number of individual actin molecules that fuse together in such a way that they form a very strong filament. This filament is the functional form of actin. Here we see the protein code of a virus. Each of the small globular units is an individual polypeptide chain that forms a sphere by itself, but these spheres of polypeptide join together to make this protein coat, as we see here. Collagen and elastin are structural proteins of the extracellular matrix. Collagen gives a tremendous amount of strength to the tissue, whereas elastin allows the tissue to stretch and then recoil to its original shape. Both types of structural proteins consist of many subunits joined together in a very organized way, and that organization actually contributes to a higher order of organization in order for the function to take place properly. Proteins in cells do their jobs by binding to other molecules. The molecule that a protein binds to is generally referred to as its ligand. The ligand may be a molecule of any different type that must bind to the protein. The binding, however, is non-covalent, so it consists of weak interactions, such as we've talked about earlier, so that that binding can be reversible. Here we have a schematic of a protein colored green, and its binding site is occupied by its ligand, which is colored yellow. The folded structure of a protein's binding site positions amino acid side chains in exactly the right positions to be able to form many electrostatic interactions with the substrate according to the exact shape of that substrate. In this example, we're looking at an enzyme, and the enzyme has various residues that can form hydrogen bonds and other electrostatic interactions in positions that correspond exactly to the substrate, cyclic AMP's structure and orientation in that binding site. Antibodies are proteins produced by your immune system to fight disease. Each different kind of antibody targets a particular disease-causing agent, an antigen. Each different kind of antibody has two binding sites for that particular antigen that it targets to immobilize. Enzymes are divided up into different classes. Each different enzyme class is a group of enzymes that perform the same category of reaction. As we can look at table 4-1, we see some examples. There's no need for you to memorize these different examples. What I want you to see is merely that there are many different kinds of enzymes, but we have a way of categorizing them. This is a schematic of some particulars about how an enzyme works. If we follow the illustration across from left to right, we see a substrate illustrated in red and an enzyme illustrated in blue. We begin with the encounter between the substrate and the enzyme, still separate from one another. If we move to the next step, the second view, what we see is that an enzyme substrate complex has formed. The enzyme has the substrate bound in its active site. The third picture across the way would be when the enzyme has catalyzed the reaction and now the product is in the active site of the enzyme. And then finally, all the way across on the right, what we see is that the enzyme has released the products of the reaction. The enzyme looks identical to the way it looked on the left, and so it would be able to act upon a new molecule of substrate. Our schematic actually was an illustration of this particular enzyme, lysozyme, which we'll use in lab in a few weeks. Lysozyme has a binding site that holds its substrate, as we can see in red in this illustration. In a chemical reaction that does not involve an enzyme, the thermal energy of the substrates within the cell and random diffusion must bring the substrates together in order for the reaction to occur. Not only that, When the substrates come together, they must be in the correct orientation, and they must collide with enough force that the particular bonds that have to be broken or that need to be made must be strained. For a chemical reaction that's mediated by an enzyme, much of the randomness that's necessary for a non-catalyzed reaction is overcome. For example, the enzyme will promote perfect orientation of the substrates, a precise orientation that encourages the reaction to take place between them. Also, the binding of the enzyme to the substrate will rearrange electrons in the substrate so that partial negative and partial positive charges are formed in just the correct places. This binding promotes a change in the shape of the enzyme itself. That shape change will strain the bound substrate molecule. It will put stress on the chemical bonds that must be rearranged and put it in a very unstable state that we call a transition state, which favors the continuance of the reaction. So this explains how an enzyme is able to lower the activation energy for a reaction. It actually provides a different mechanism for a reaction as compared to an uncatalyzed reaction. All of the factors, the precise orientation of the substrate, rearranging of electrons in the substrate, and changing the shape of the substrate to the strained transition state will contribute to the low activation energy of the reaction when it's mediated by the enzyme.